Welcome back. This is the fifth module, enzymes and coenzymes. And here we are going to learn about the classification, nomenclature system, and properties of enzymes. Then the mechanism of enzyme action. We will also be looking into the active center. Then theories of enzyme action, enzyme inhibition, and then the coenzymes and cofactors. Okay. So what are enzymes? Before we go into the detail on enzymes, enzymes are basically biological catalysts. Okay. So what are catalysts then? Catalysts are any substance that increases the rate of a chemical reaction without itself being changed or consumed in the reaction. So a catalyst is a substance which can increase the rate of a chemical reaction. That is, if a particular reaction A is giving maybe like A plus B giving C plus D is a reaction, the rate at which the particular products are formed from A and B, the reactants, it will be increased. Okay, the rate at which means the within a particular time how much product C and D are produced. Okay, so that will be increased with the help of a catalyst. Fine. What happens to the catalyst during the reaction? Obviously, the reactants A and B will get converted into the products C and D, but the catalyst, they remain unchanged. They won't get consumed during the reaction. They won't get modified during the reaction. Okay, so that is specific. So enzymes are biological catalysts. So enzymes also play a role of catalyst in biological systems. Okay, and each enzyme, they catalyze specific set of reactions. You can see that these compounds on which the enzyme act upon, they are known as substrates. Okay, as we mentioned, reactants giving rise to products. Here we have the substrates. Okay, so enzymes actually act upon the substrates to yield the product state. Okay, now there are enzymes are basically proteinaceous in nature, except for the RNA that acts as enzyme in the case of ribozyme. All the enzymes are colloidal, thermolabile in character, in the sense they are actually influenced by the increase or decrease in temperature. The enzyme action is influenced by the temperature range. Okay. Then, when we discuss about the enzymes, so we have already seen they are biocatalysts which are synthesized in living cells, and we also saw that they are protein in nature except for ribozymes. They are colloidal and thermolabile in character, and they are specific in their action. So each enzyme do have exhibit specificity with respect to the substrates upon which they act upon. Okay, so when we look into the historical background, the term enzyme was introduced by Frederick Kuhn in 1878, and he used this term to indicate the catalysis taking place in the biological system. And it was in 1883 Edward Buckner isolated the enzyme system from cell-free extract of yeast, and that is about the historical background. Okay, now depending upon the functions they perform, depending upon how frequently we come across a particular enzyme in a biological system, we can find different groups of enzymes. Okay. The first one we can see is the constitutive enzymes and inducible enzymes. So what are constitutive enzymes? Constitutive enzymes are otherwise known as housekeeping enzymes. Okay, that is, constitutive enzymes are those enzymes which are present in a cell throughout in every cell. Okay, and or the concentration almost remain constant throughout. Okay, so those are known as constitutive enzymes. For example, cytochrome oxidase. Cytochrome oxidase is an enzyme that is involved in the cellular respiration, and that is required by every cell that actually takes up respiration, isn't it? So it is present in all the cells, and the concentration, it has to be kept almost constant throughout. Okay, so such enzymes are known as constitutive enzymes, or otherwise it is referred as the housekeeping enzymes. Okay, now the second one is known as the inducible enzymes. In the case of inducible enzymes, the concentration of enzymes increases when there is enough amount of substrate present. Okay, so it increases or decreases as per the substrate concentration. So The concentration of inducible enzymes is almost directly proportional to the substrate concentration. So the presence of substrate induces the production of enzymes in a cell. Okay, for example, lactate dehydrogenase. Okay, the lactate dehydrogenase is an enzyme that acts on the lactic acid. Okay. Um, Okay. So the lactate dehydrogenase is an inducible enzyme, and the enzyme, the lactate dehydrogenase, is produced in the presence of lactic acid or lactate. Okay. So it is involved in the interconversion of pyruvate and lactate. The lactate dehydrogenase enzyme is catalyzes the interconversion reaction of lactate and pyruvate, and it is an inducible enzyme. The enzyme production is like under the influence of the concentration of the substrate. Right. Now another concept is regarding the Abzymes. Okay, this particular term itself comes from the antibody enzymes. Okay, and they are catalytic antibodies which can function as enzymes to catalyze specific reactions. So they are basically antibodies, and you all know that antibodies are basically proteins. So they are also proteinaceous in nature. Okay, so catalytic antibodies are referred as the Abzymes, and these are examples for inducible enzymes. They are produced only in response to the presence of specific antigen molecule. Okay, so this is so we have found two sets of enzymes based upon when these how frequent these enzymes are present in a cell, and how like what are the functions. Okay, then we saw Abzymes, which are antibody enzymes. Okay, another concept is regarding the zymases. Okay, zymases and zymogens. Zymases are active enzymes, that is, when they are produced, from the time it is produced, the enzymes are active. Okay, and these are the glycolytic enzymes. Glycolytic enzymes are required by the cell whenever energy is required. So you can see that glycolytic enzymes, all those enzymes involved in the glycolysis process, okay, they are active enzymes, and they are called as referred as active enzymes. Okay. Um, Here you can see that they can act directly upon their substrates as and when they are produced. But what about zymogens? Zymogens are those enzymes which are produced in inactive form. Okay, inactive precursor form. Fine. And only when these enzymes are required for the catalysis of the reaction, it becomes activated. Okay, so some enzymes secreted in their inactive forms are referred as the zymogens. Okay, or they are also referred as proenzymes, right? And they are made actively functional by certain activating enzymes or agents. That is, only when there is a specific condition present in the medium, the zymogens become activated. Okay, so these zymogens become activated, and during this process they undergo a irreversible activation by the breakdown of one or more peptide bonds in the zymogens. So in the zymogens, when they are influenced by or activated by certain agents, what happens during that process? Certain peptide bonds it get broken, and this will result in an irreversible activation. Okay, so once zymogens are activated to form the proper active enzymes, it cannot go back, or it cannot become zymogens later. Okay, so this is a irreversible activation. There are plenty of examples from the digestive enzyme set. For example, chymotrypsinogen, it is a zymogen, and which when activated, it gives rise to chymotrypsin. Okay, it is a protease. Fine. Pepsinogen, pepsin. Okay, pepsin is again protease present in the gastric juice, and this is actually present in the zymogen form or precursor form, pepsinogen. Similarly, plasminogen. Okay, it gets converted to plasmin. So pepsinogen, it is activated by the hydrochloric acid. Okay, when hydrochloric acid, or when the medium inside the stomach, it becomes acidic enough, the pepsinogen, it becomes pepsin. Okay, similarly, trypsinogen, trypsinogen to trypsin, the conversion is actually controlled by the enzyme enterokinase, and chymotrypsinogen is conversion to chymotrypsin when there is enough amount of trypsin in the intestine. So all these are digestive enzymes we have discussed. Okay, these are the extracellular digestive enzymes: pepsin, trypsin, and chymotrypsin. Clear? So zymases are those enzymes which are produced, which when produced are active. Okay, they can directly act upon the substrate. All the enzymes involved in the glycologic process, they are zymases. Okay, zymogens are precursor forms. Okay, when they are produced, they are in the inactive forms. Okay, only when it is being acted upon by specific activating agents, the zymogens get converted into the active enzyme form. Okay, and usually during this conversion of or activation of zymogens, what happens is one or more peptide bonds are broken in the zymogens, and this will result in an irreversible activation of the zymogens. Fine. And there are plenty of examples of zymogens from the digestive enzymes set. For example, chymotrypsinogen, trypsinogen, pepsinogen, etc. Okay, chymotrypsinogen is a zymogen of the active enzyme chymotrypsin, which gets converted. I mean, which actually gets activated. Chymotrypsinogen gets activated to chymotrypsin in the presence of sufficient amount of trypsin in the intestine. Okay. Now, pepsinogen gets converted to pepsin, or pepsinogen gets activated to pepsin by the presence of acidic medium, or in the presence of acidic medium because of HCl in the stomach. So this is regarding the zymogen and the what you call zymases. Okay. Another set of classification is based upon the site where the enzymes act upon. Okay. One is the intracellular enzyme and the extracellular enzyme. Right. Intracellular, the name itself indicates it is inside the cell. Right. It is otherwise referred as the endoenzymes. So when the enzymes are functional within the cells where they are synthesized, they are referred as the intracellular enzymes or endozymes. Okay. Example: cytochrome oxidase. Cytochrome oxidase is an enzyme which is required for cellular respiration, and it is present inside the cell, or it is functional inside the cell. Another is the extracellular enzyme. Extracellular enzyme is otherwise referred as exoenzymes, and these enzymes are active outside the cell. Okay, so these enzymes are produced in the cell. It is then released out of the cell, and it is active and functional outside the cell. Okay, so such kind of enzymes are known as exoenzymes or extracellular enzymes. And these enzymes, we have sufficient examples, and all the digestive enzymes belong to the exoenzyme category, that is amylases, proteases, lipases. All these are produced inside the cell, one or the other cells. Okay, but they act on, they become functional only outside the cell, isn't it? It is in the digestive tract. It is not inside the cell, it is outside the cell. Okay, so that is why it is referred as the extracellular enzymes. Okay, so these are the different categories or groups of enzymes based upon various criteria. Enzymes are divided into 6 major classes, and that kind of classification is mainly what you call based upon the chemical aspects and the functional aspects. Okay, as a little more scientific way of classifying enzymes. Okay, and these are the 6 major classes based upon what kind of reaction they catalyze. We have 6 categories: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Taking the first letters of all these, we have OTH, LIL, and that is a mnemonics we can use for learning the 6 categories in the sequence, because the sequence have to be maintained. Okay, so the detail we'll be looking into in the next presentation, the classification of enzymes as well as the nomenclature system. Okay.